Hello, everyone. My name is Borut Kovacic. I am senior embryologist, the head of IVF laboratory in University Medical Center Maribor from Slovenia. I'm also the professor of cell biology at the University of Ljubljana. And I will talk about new frontiers in sperm selection for medically assisted reproduction. I declare no conflict of interest, and the following objectives will be discussed. First, I will explain what is the principle of sperm selection in a natural fertilization. Then we will talk about the conventional sperm selection techniques for medically assisted reproduction. I will focus then to optional sperm selection techniques for MAR that can be categorized in the following categories, like microscopy-based techniques, binding assay techniques, sperm membrane potential-based techniques, the methods that are based on the swimming characteristics of sperm, many customized techniques that we modified according to the severity of male infertility, and the last couple of years there are some studies that show very promising artificial intelligence-based sperm selection techniques. And let's go first to the principle of sperm selection when they enter the female reproductive tract. The first sperm selection filter is definitely cervical mucus. It is known that the architecture of mucus represents a filter, and there is just limited number of days when the sperm are allowed to swim through the cervical channel. The sperm there are selected according to morphology and motility, and once they reach the uterine cavity, they meet leukocytes that start with phagocytosis, particularly to sperm that are not protected with seminal plasma. So it is also interesting that the temperature within a uterine cavity is a little bit lower than the temperature in the oviduct, and the sperm have a tendency to swim from cooler to warmer places. So the oviduct is a kind of sperm reservoir. It comes to transient adherence of sperm to epithelial cells that are attached with sperm rostral proteins, and the epithelial cells modulate the capacitation of sperm. In fact, they release the sperm in limited numbers to swim toward ampular region where the fertilization should occur. So this sperm selection filter select the sperm according to membrane integrity and DNA integrity. And once they reach the cumulus of forus, this represents the chemoattractant for sperm. It is derived from follicular fluid, and the sperm are selected according to motility, hyperactivation ability, also intact acrosome, and DNA integrity. And the final checkpoint, the final selection filter is definitely zona pellucida. Zona pellucida is the most stringent filter for sperm, especially the zona pellucida 3 protein that represent the place where the sperm attached to zona pellucida. And as we can see on this immunofluorescent image, only morphologically optimal sperm can attach to zona pellucida, and they're selected according to capacitation, acrosome intactness, superior morphology, and DNA integrity. Let's go now to sperm selection techniques that are used in medically assisted reproduction. There are 3 conventional methods. The first one that represents the golden standard is a density gradient centrifugation, where the sperm are selected on barriers between different viscous columns of silica gel medium, and only motile and morphologically optimal sperm are able to pass these barriers. So when this centrifugation is ended, then the semen sample cannot be used as such because the sperm sample must be washed from silica gels. And there is another centrifugation needed with the sperm preparation media and after this centrifugation, only the sperm can be used for IVF. So the next method is a conventional swim-up method. This method also includes the centrifugation, where the small amount of ejaculate is resuspended in a sperm preparation medium. The tube is then centrifuged. The pellet, the supernatant is rejected, and the pellet is overlaid with the fresh sperm sample, with the fresh culture medium, and the motile sperm are allowed to swim up in a fresh culture media. The third method is a method without centrifugation. It is one of the oldest method called underlay method, where the small volume of ejaculate was simply placed on the bottom of the tube with the culture medium, and the motile sperm are allowed to swim up in upper layers of culture media. Very similar principle is used in commercially available, we are calling microfluidic devices, but in fact this is not a completely microfluidic device. The method could be called side lay method because the sperm ejaculate is injected in a inlet on the side position of this plate, and the sperm are then swimming through the channels and through the grid to the culture media, and this larger opening represent the outlet where the semen samples can be used then for MAR techniques. So the main principle of all these conventional sperm selection techniques are to isolate progressively motile and morphologically optimal sperm from semen samples. Like we can see here, this is a semen sample with a high concentration of sperm, but also some epithelial round cells and many immotile cells and debris. Once this sample was processed with one of these techniques, the semen sample appeared as this one. No debris, no immotile sperm. The motile sperm are hyperactivated, and this sample can be used then for insemination of oocytes or for intrauterine insemination. The conventional sperm selection techniques have also some drawbacks. Which are these drawbacks? The first one is the utilization of various centrifugation steps and exposing of healthy sperm to the pellet, to debris, to leukocytes, where there is a high level of reactive oxygen species that can cause oxidative stress in sperm cells. The centrifugation itself can decrease the motility and can cause damages in sperm membranes, particularly in sperm tails, and also can cause DNA damages. In swim up of semen in severe male factor of infertility, also in some moderate oligozoospermia, where there is a need for additional centrifugation or concentration of motile sperm, such samples are often poorly washed from seminal plasma and debris. And it should be known that only 55% of injected sperm used for ICSI contain normal DNA. So why should sperm selection techniques for MAR be improved? First, because the female reproductive tract is very selective for sperm, as it has been presented in the first slide, and only a small fraction of morphologically superior sperm out of millions of them can reach the oocyte in ampular region. The male infertility is a factor in only 30 to 50% of infertility couples. This means that approximately 50 to 70% of all IVF cycles should be conventional IVF cases, not ICSI cases. And about 40 to 88% of sperm samples from infertile men have high level of reactive oxygen species, which decreases sperm motility and DNA integrity. And currently employed sperm selection techniques completely neglect other important factors such as DNA integrity, reactive oxygen species generation, membrane maturation, and non-apoptosis. They are based only on their own sperm motility and morphology. So let's see now what are optional sperm selection techniques for MAR. There are 6 groups. The first one is microscopy-based techniques. I will try to explain all these optional sperm selection techniques that can be found in the literature. The second one is based on the binding characteristics of sperm. There are so-called PIXI, Max techniques and zona binding techniques. Then there are membrane potential-based techniques, 2 of them, not a lot of publications about these methods. And then very interesting swimming potential techniques. I will explain different properties of sperm during the swimming. Some other intervention techniques, in fact, these are customized techniques, and then the 6th group of artificial intelligence-based techniques. So let's start with the first one. What is the background of microscopy-based techniques for ICSI, for example? We should know that most sperm contain 1 or more vacuoles that are not easily visible at the magnification at which we perform the ICSI procedure. And this was something that led the scientists to develop the new methodologies by exploring ultra-high magnification of microscopes that at 6,000 times enables to perform motile sperm organelle morphology examination with the aim to see the vacuoles in sperm heads. And the second very interesting phenomenon is that we are talking about double refraction or birefringence. This is a decomposition of light into 2 rays when the light passes through an anisotropic material. And exactly this phenomenon is a characteristic of cells with normal nuclear organization, where the nuclear protein filaments are oriented longitudinally. So there are two methods. One is polarized microscopy, exploring birefringence, and the second one is so-called IMSI method. Regarding birefringence, the first study was performed by Sismer group, and they showed that the sperm heads that showed the birefringency are in fact reacted spermatozoa, and they compared the clinical outcome in 2 groups. The first group where the reacted spermatozoa were used for ICSI, and the second group with non-reacted spermatozoa. The study was performed on a very small number of cycles, but it has been presented that the clinical outcomes were significantly improved by reacted spermatozoa. What about IMSI? When this method appeared, it was a lot of studies presenting nice images with the increased sperm heads, and the arrows usually show the vacuoles in the head or in the nuclear region. And most of these first studies presented significant advantages and improved clinical outcome when this method was used for selection of sperm for ICSI procedure. But the first meta-analysis performed by Belgian group showed that techniques was not effective for any unselected ART patient. And for severe male factor, the evidence suggested a higher pregnancy rate with IMSI. However, the benefit for repeated ICSI failure patients remained unproven. In the same year, the Cochrane Library Group presented the meta-analysis including only RCTs, and the results from this analysis didn't support the clinical use of IMSI. They concluded that there is no evidence of effect on live birth or miscarriage, and the evidence that IMSI improves clinical pregnancy is of very low quality. The second group of methods are based on binding characteristics of sperm. The zona pellucida bound spermatozoa are morphologically superior, was presented particularly by Liu and Baker in the period before ICSI. And the second very interesting study was from Husar and colleagues. They showed that mature spermatozoa may bind to solid-state hyaluronic acid, and spermatozoa with advanced levels of capacitation or acrosome reaction don't exhibit this hyaluronic acid binding. So the third study is also very interesting. The author showed that apoptotic spermatozoa express the phosphatidylserine in their membranes, and annexin 5 conjugated microbeads can attach on the phosphatidylserine and are then exposed to a magnetic field which separates apoptotic cells from non-apoptotic ones. So there are three different methods. One is zona binding, physiological intracytoplasmic injection of sperm, so-called PIXI method, and by using PIXI Petri dishes, and magnetic activation cell sorting. And let's see the details of each of these three techniques. The zona pellucida bound spermatozoa showed when they're used for ICSI, the authors showed the high quality embryo rate and the higher embryo transfer rate in this study than in the control group. The second study showed very similar results, but we can conclude that there are only few studies, all with very small number of cases, and practically it is very difficult to implement this method in clinical work, because then some oocytes should be used just for selecting embryos, and the timing can be problematic because all oocytes have a limited time when they have to be inseminated. So the method today is not evidence-based. What about hyaluronan-bound spermatozoa? These images nicely showed in A image that when the sperm are placed on the slide with the hyaluronic acid, we can see all kinds of sperm that are hyperactivated with already reacted acrosomes. But when this slide was washed, only the sperm with intact acrosomes and DNA integrity remained attached on the hyaluronic acid. And on this principle, the PIXI dish is working. So this on the right side, there is the first clinical study that shows that in couples where less than 65% of sperm bound hyaluronan, the selection of such bound sperm for ICSI led to a statistically significant reduction in pregnancy loss rate. but there was no difference in implantation and pregnancy rates. So there is another study. This is the largest randomized trial on pixie dish to date, and the first to provide a robust measure of live birth. But it showed that pixie didn't significantly increase the term live birth rate compared with standard ICSI, but a significant decrease was observed in miscarriage rates among couples in the pixie group. So regarding magnetic activated cell sorting, or so-called MAX techniques, there are many papers that presented the benefit. The conclusion of all these techniques can be that MAX is a method for selection of viable sperm with low DNA fragmentation index when applied to raw semen, especially in samples with low values of progressive motility and vitality, and it helps to decrease miscarriage rate. But later, the same number of papers can be found presenting completely no benefit. So the summary of these papers shows that integrating MAX as a part of sperm preparation techniques will not improve sperm fertilizing potential to the same extent as the traditional swim up or or other conventional sperm preparation, sperm separation procedures, and the separation with MAX caused a significant loss in the number of total and rapid progressive spermatozoa. So if it is used, then it cannot be used for oligozoospermic sperm samples. The membrane potential-based techniques, we should note that human sperm plasma membrane hyperpolarized during capacitation, and this is necessary for the acrosome reaction to occur. Potassium channels are involved in this hyperpolarization, and based on these characteristics, there are two methods. One is microelectrophoresis, and the second one is very simple zeta potential method. The microelectrophoresis method, this is only one paper, and in this pilot study, it was demonstrated that membrane-based electrophoresis is as effective as density gradient centrifugation method, although it takes only a fraction of the time. The simple zeta method for sperm selection, that is based on sperm membrane charge, is the principle of this method is so that live sperm should be washed in serum-free Hippus medium that is then negatively charged. And to ensure that the tube is positively charged, the tube should be placed inside the latex glove, rotate 2 or 3 times, and rapidly pulled out. So the results of this paper showed significantly increased percentage of forward progression, hyperactivation, DNA normal integrity, strict normal morphology, acrosome intactness, not only in male group of infertility, but in all inclusive groups. So the swimming potential techniques, it is very interesting to know, and it is not very well known, that human spermatozoa have a tendency to touch during swimming. This phenomenon is called thigmotaxis. They also have a tendency to swim against the stream of fluid. The phenomenon is called rheotaxis, and to swim from cold to warm places. This is thermotaxis, and also toward chemoattractants released from cumulus corona oocyte complexes. This phenomenon is called chemotaxis. So there are a couple of methods based on these sperm swimming characteristics, and let's go one by one. One of the first microfluidic device was presented here. You can see it on the right side. It is like a small coin, but in fact, it is from two set of channels. There is a tubing system connected to both sides, and the capillary force driven pump was designed to maintain a steady flow rate of approximately 8 nanoliter per second. In the upper gray highlighted channel, this is a place for native semen sample, but in a white channel there is a culture medium, and as it can be seen here, the motile sperm can swim from native semen spontaneously to the fresh culture medium. So the results showed that the proportion of motile sperm is significantly increased when they're taken from outlet. After this spontaneous passing of spermatozoa from native to culture medium. There is another very simple method exploring rheotaxis. The normal Petri dish was designed with different droplets. The red one is the polyvinyl pyrrolidone that is used for ICSI. The blue one is a native semen samples, and the yellow one are the culture media. They are then connected with the fluid bridges. And if we follow what is happening with the fluid, there is a very tiny fluid flow from the more viscous to less viscous medium, and the sperm show the tendency to swim from the native semen samples toward PVP. So the clinical results by using the sperm that are selected by this way show the significantly increased sperm morphology and the lower number of abnormal chromatin condensation. This is the very similar techniques with not commercially available microfluidic device. In fact, there is a plate with a set of different channels, and the sperm are allowed to swim through this channel, and they are selected based on the pigmotaxis. The sperm from the outlet showed the high DNA integrity level. This is very interesting experiment. The slide was designed to simulate the female reproductive tract with two oviducts, and on the right side the authors placed the cumulus cells, and they were looking what is the proportion of sperm cells that decided to swim on the right side and not on the left side. And around 10% of human sperm showed chemotaxis. It was 10% more sperm found on the right side where the cumulus cells was positioned. The next study is also very interesting. A special micro device was designed that was heated on the right side, and there was a difference in the temperature, with a lower temperature on the left side and higher temperature on the right side. The set of experiments were performed, allowing the sperm to swim from cooler to warmer places and opposite, and these sperm were then used for ICSI in mouse oocytes and also in human. They showed that such selected spermatozoa in mice and humans show a much higher DNA integrity and lower chromatin compaction than unselected sperm. And at least in mice, when the ICSI was performed with the sperm that had the capacity to swim from cooler to warmer place, these experiments gave rise to more and better embryos through ICSI, doubling the number of successful pregnancies. What about some other interventions that are needed when we have very complicated sperm samples? I call this like customized methods, and there are different dependent on the semen quality. When we have the cryptozoospermia with just few spermatozoa in the whole ejaculate, usually we do the centrifugation of the whole sperm sample, and then in a micro droplet we are looking for the sperm that are able to swim out of very contaminated droplet with the debris and some other cells. So in fact, this is spontaneous selection of sperm that are able to swim out to the PVP. The leucospermia is a problem with a large number of round cells, and if we use the conventional gradient, then it can happen that these cells can block the sperm to pass through the barriers between the lower and the higher concentration of silica gels. So in such cases, it can be that the sperm sample is centrifuged, and immediately after centrifugation, the sperm are mechanically isolated by the micropipette to the PVP, where they're washed by changing the position in the PVP droplet and then used for ICSI. Regarding globozoospermia, this is a phenomenon where the sperm have no acrosome, but this total globozoospermia is very rare. Mostly we can find partial globozoospermia with at least couple of sperm with the intact and normal acrosomes. So what to do? In fact, first we have to be persistent in searching sperm cells with acrosomes, but if we can not find these such sperms, then we have to use some activators that can be chemical agents, or we can use mechanical methods or electrical stimuli to activate the oocytes. There are a couple of papers that show how this can be performed. Absolute asthenospermia is the case with no motile spermatozoa. There are different reasons for such situation. It can be necrozoospermia, but it can be also the defect in the motor apparatus, and also testicular sperm are usually totally immotile. What are possible interventions? In fact, the most simple one is to use the sperm motility stimulator. There are pentoxifylline, theophylline, many others, and many publications that show the positive effect of this sperm motility stimulator and how the method of viable sperm cells can be identified and isolated. The hyposmotic screening test has many false positive reactions, but it can be used also in specific cases. Then there is a sperm tail flexibility test, and in TSEP, if we cannot find the motile sperm also after adding the sperm motility stimulator, and if we get the very low fertilization rate, then some authors recommend to use the testicular sperm. If this is a phenomenon found in the ejaculate, then they recommend to use testicular sperm in cases of necrozoospermia. So sperm tail stump defect is a phenomenon where the sperm tail is not developed well. It is very short and thick one. And what can we do? Because it is very difficult to recognize which sperm are viable. We can use the sperm without showing any motility. But if we use the hyposmotic screening test, then as it can be shown on the right side, some vital sperm can be recognized by the swollen sperm, short sperm tail. Regarding testicular sperm, there are different methods described. Usually there are mechanical methods with shredding, fine mincing, vortexing, and crushing in grinder of testicular tissue. But there are usually then mechanical isolation of sperm from this cell suspension with ICSI micropipette. Usually there can be some optional additional centrifugation or additional sperm motility activators, erythrocyte leasing buffers, or some enzymes like collagenase to make cell suspension from the tissue, and some laser-assisted immotility sperm selection. There is a presentation how we can help ourselves when we have very hemorrhagic testicular sperm sample, especially if we are doing testicular sperm aspiration. There are only erythrocytes found, but if we add the pentoxyphylline or theophylline, then the sperm start to move, and we can recognize the place where we can find this sperm. The whole group of erythrocytes with this moving sperm can be then aspirated in the ICSI pipette, and the cells are then expelled to the PVP, and the sperm can be then isolated from other erythrocytes. Usually we do the additional sperm selection in the PVP, especially on the margin of this droplet, because there is possible to see nicely the sperm head, and we can deselect the sperm having large vacuoles. What about the artificial intelligence-based methods? There are a couple of publications nowadays. Usually there are the methods that try to select the sperm by morphology and motility, and there are creation of different artificial intelligence models and softwares that can be used for better selection of sperm for ICSI in real time. So we have here two publications. The first one in this slide, I can present the selected study from 2022. And according to the authors, this is the first study to implement automatic real-time determination of the morphology of swimming spermatozoa using machine learning. To create this new deep learning algorithm, the authors generated a unique set of more than 4,600 images that served as input data for YOLO3. What is YOLO3? This is a popular real-time object detection algorithm, and the images were also used as the training data set for the proposed algorithm. The authors performed a validation, and the results indicated a high sensitivity and positive predictive power of the model for detection of abnormal and normal shaped swimming spermatozoa, and slightly lower predictive power in the case of vacuole detection. So the top right image displays a heat map used to visualize how the deep learning model makes its predictions. If it was a spread of signals, then the model was not able to completely recognize abnormal from abnormal sperms. But where the warm color signals were concentrated around the head, then these sperm were recognized or as normal or as abnormal. So the developed artificial intelligence algorithm has good potential as a tool to assist embryologists in selecting the optimal sperm for ICSI. Then there is another study. This is computer software, so-called SID or sperm ID. It has been developed to assist in selecting individual sperm for ICSI. And this SID detects motile sperm and analyzes each sperm in the visual field using a digitizer attached to an ICSI microscope. But the aim of this study was to test whether there is an association between individual sperm kinematics measured with this SID and the sperm selected by an embryologist with subsequent outcomes, such as normal fertilization and blastocyst formation. So the results showed statistically significant differences in SID scores between spermatozoa leading to positive versus negative fertilization outcomes, as well as between those associated with blastocyst generation and no blastocyst generation. On the lower image, it is possible to see the green circle that, where the model spontaneously select the morphologically normal sperm, and this system can help embryologists to select the more optimal sperm for ICSI. So we can conclude that density gradient centrifugation or swim-up still represent the gold standard for sperm selection in assisted reproduction technologies. If we have some difficult cases, then we need to customize sperm preparation methods. And although efforts have been made to develop more efficient techniques to select optimal sperm for IVF and ICSI, data are still insufficient or controversial to conclude that they are superior to sperm density gradient centrifugation or swim-up. And although most of these novel methods were still not applied in routine art or have not been tested enough, the results are very impressive as they tend to mimic physiological conditions. So, thank you very much for your attention.